thank you everyone for joining and for inviting me to the organizers. It's a real pleasure, especially to talk to audience, with a similar or even overlapping passion. So I know that I don't have to explain a lot about the motivation. I will just go ahead and share my slides. I hope that works fine. And okay, I have my slides on the screen next to this one. So I hope you don't mind me looking a bit sideways. I'll try to be with you and we go over questions, etc. So I am going to be talking about one specific area of our research today, which is protein evolution, specifically focused on structure evolution during life's early evolution and origins. So basically, mostly before LUCA is the window that we'll be looking at. The reason is we know a little bit about LUCA and what we can expect there. By LUCA, I mean the last universal common ancestor. By work of actually many of you here and others, there has been quite an advancement of some of the top-down approaches to studying how protein structures and functions evolved all the way to LUCA, or how they basically emerged from this simpler organism. But what came before LUCA is very hard to approach using these top-down approaches, and we're basically left with synthetic biology, synthetic chemistry, and these bottom-up approaches, and a huge portion of philosophy, I would imagine. So the question we are asking here is basically, all these nice folds that we see today in nature, and some of them going all the way to LUCA, how were they accessible by early life preceding LUCA, and how did they come about in the first place? And I will talk a lot about how this depended. I don't know if you see my mouse. Let me just make sure that I have some pointer here. Okay, and how this relates on the other side to the components, to amino acids that actually make up proteins as we know them. So the components that our life uses today is mainly these 20 canonical amino acids. You can argue, well, that's not it. We have a bunch of post-translational modifications. We also have the 21st and 22nd amino acid. But these 20 is basically what all life shares, all life without any exceptions known to us. So basically we can say our life really depends on these 20 amino acids that make up the modern genetic code. We know that this code has been developing. It is still developing. We know organisms where there are differences into the coding mechanisms, but more or less this has been established ever since the last universal common ancestor, as far as we know. At the same time, this means that this somehow was formed during the first 10 to 20% of our Earth's history, which to me is pretty amazing, that on the scale of the whole history of our life, this actually got fixed very early on and remained mostly unchanged ever since, which tells us something about the optimality of this alphabet and the need for its further adaptations. So we're going to be looking at this kind of transition from chemistry to biology, where the amino acids have been selected. And one of the first things that I would like to talk about is that only about half of today's alphabet, we call them early amino acids, were apparently around. We're apparently around easily using sort of the prebiotic synthesis. And this has to precede the time of LUCA. Actually, by the time of LUCA, we think we already had the full alphabet around. At the same time, the late amino acids, which are not shown here, probably came around by biosynthetic pathways, which already required some form of chemical networks and metabolism to be functioning. How do we know this? Well, of course, we don't know this for sure, because this is very far back in time, and we're kind of left with very indirect interference here. How the early alphabet has been denoted was based on a number of studies focusing on both how amino acids could be formed from prebiotically abundant chemicals on the earth and how they could be delivered from exogenous sources such as meteorites. And quite astoundingly, we do see an overlap from both of these general sources of prebiotic material, showing that about these 10 really do come up quite constantly in these sources. I'd just like to point out that these are generally the smaller amino acids from our alphabet, kind of agreeing with that they are the easier ones to make. And quite interestingly, we do see the two canonical negatively charged aspartic and glutamic acid, but we do not see the positively charged amino acids among them. And we don't see, for example, aromatic amino acids, which are very important, at least from what we thought, for forming protein structures and holding proteins together. Of course, I'm making this super simple because prebiotic chemistry is anything but clean. And we know that there were many other amino acids found prebiotically. We call them non-canonical amino acids or non-biological amino acids. Those that were around from the exogenous or endogenous sources, but for some reason didn't make it into our protein alphabet, into our biological protein alphabet. These are just examples of such amino acids, but one of the biggest questions for us protein biochemists being, why didn't they make it into our protein alphabet? Why did our life choose these 10 early ones, or keep them, and why then wait for these 10 late ones to be incorporated into genetic code only after their synthesis was somehow available? So this is one of the first questions that I'm actually going to talk about, covering sort of this very early era of Earth history. I'm going to cover hopefully two topics today. The first one will focus on peptides, so the very small versions of proteins, peptides that could be synthesized prebiotically by different prebiotic synthesis pathways, and kind of looking at them as the biggest ancestors of proteins, the longer sequences that today make up those nice structures. And only after talking about peptides, the kind of first polymers made of amino acids, we're going to switch to early proteins, so to small proteins that could actually be the first, resemble the first protein structures like we know them today. So as I said, one of the first questions that we had surrounding the prebiotic chemistry and what we know about it was, why did our life choose those 10 early amino acids into the coding, and why not some other non-canonical amino acid? So we decided to do a very, very simple experiment, and we basically mimicked peptide sequences that we could get from these different sets of amino acids. I'm going to show you a very simplified version of what we did. So I'm going to talk about 4 such sets of sequences. and they will be made from these different subsets. So what they all have in common is 7 early amino acids that kind of always appear among the early set. And then I'm going to talk about 2 canonical versions of our alphabet, the 10 early that I just talked about, and the 10 early plus a representative of positive charge, arginine. So these are representatives of the canonical amino acids. And then I'll compare them with 2 non-canonical sort of counterparts to these. So the green one, called 10 unbranched, is simply where a library where we exchange isoleucine, leucine, and valine, which are aliphatic branched amino acids, with aminobutyric acid, norvaline, and neuroleucine, which are very, very similar amino acids if you look at them. They're also aliphatic. The only difference is that the side chain is very linear. It's not branched like those amino acids above it. And these were not selected into our alphabet, and we want to kind of compare, why were these left behind and why did we choose the branched amino acids? And then on the right, the yellow library is one where we add also a positive charge to the 10 early alphabet, but this positive charge is non-canonical. It's deaminobutyric acid. It's one of the amino acids that was prebiotically available according to many different experiments and sources. It has a short chain, very much unlike, for example, arginine, which was selected into our proteins. And we wonder why didn't nature stick to this? And so Misha, our former PhD student, basically synthesized these 4 peptide libraries. These libraries were made by chemical synthesis, and they each contain very random sequences. So basically we do have 4 test tubes. Imagine we have 4 test tubes, and in each tube we have 1 library where we don't have 2 sequences, 2 same sequences. Each sequence, statistically speaking, is different. It's super combinatorial and very large sequence space. And we are interested in kind of the collective properties of these individual libraries, of these 4 libraries, and how they differ between them. This is very simple science. We cannot use many methodologies because it's a very kind of combinatorial mixture. But one of the first things that we looked at was their solubility. We compared solubility of these 4 libraries. Again, these top ones are the canonical versions. These are the non-canonical versions. And what you are seeing is solubility across a pH range using 2 different ionic strength conditions. And without going into detail, the message I want to convey to you is that the sequences or the composition used by our proteins today is less soluble overall than the non-canonical alternatives. And you may think, solubility is a good thing. You want sequences to be soluble. So why did we choose the less soluble ones? And I can tell you already that something that we see from many different projects, especially when it comes to random sequences, that solubility is just another side of the coin with structure formation. We know that when we have random sequences and they have lower stability, that actually means they have more structure propensity, which is something we looked at here. So these plots showing the same libraries, the same 4 libraries, but looking at the CD spectra. CD spectroscopy, in case you don't use it, as a method to basically map the secondary structure potential, showing you alpha helices and beta sheet. Here again, remember we're looking at whole libraries. We're not looking at an individual clean sequence. And most of the structure content is actually not ordered. That's why we see the minimum around 200 nanometers. The places where we would see alpha helices and beta sheet is more between 210 and 220 nanometers. So the way we compared the structure content among these libraries was here in these inlet plots, where we look at the ratio of the part of the plot where structure is to the part of the plot where structure is lacking. So the higher these bars, the more structure propensity there is. So I said this in a very complicated way, but basically if you couldn't follow, what we see is that on the top libraries, the top libraries do have more structural content as expected from the solubility data versus the libraries with non-canonical amino acids, giving us a very clear message that those amino acids which were left behind, the non-canonical amino acids as we call them today, just simply were not so friendly for structure formation. And we do think that this could really represent an important property during the selection of the protein alphabet. Okay. So this was something showing us why those non-canonical amino acids were left behind, but also we started seeing that the 10E library, that the 10 early amino acids that were actually selected into our coding were somehow most prominent in many different properties. I didn't show all of them here. We also compared a few more libraries, and the 10E somehow seems to stand out. And remember, this is a very acidic alphabet. It has 2 negatively charged amino acids, no positively charged amino acids. So this was very sort of counterintuitive given the properties of today's proteins. So we just decided to look at this in a little bit more detail and compare it again with sequences of different charges. So what we did here was something very similar to like I showed you before. This time Misha compared 9 different libraries. One didn't have any charge, positive or negative. Then we have 2 libraries only with positively charged amino acids. We had lysine and arginine, like the canonical versions, and then the diaminopropionic and diaminobutyric as the non-canonical alternatives. The same case for negative charges, and then we have all kinds of combinations of positive and negative. And here, more from the sort of biophysical perspective, we were interested in, what kind of makes the 10 really special, and is it really special when we do this systematically, when we compare all the different charge scenarios that we could mimic. And okay, I'm kind of jumping fast here, already showing the arrows that among these libraries, the negatively charged ones again do stand out. They do stand out in structure formation, and here I'm showing you another plot. This is size exclusion chromatography. It's a type of chromatography where you can look at the size of your proteins. Here we are looking at peptides, and most of the libraries come out around somewhere between 14 and 16 milliliters, which is where the monomeric species comes out. This is very much expected from soluble material. We get monomeric peptides. But what we see for the acidic libraries, and for them only, there is a small peak coming very much up front at the chromatogram. So the way that size exclusion chromatography works is that the kind of biggest molecules come out early on. And so what these peaks are is some form of oligomerization. I'm going to now show you in a bit more resolution this particular library at page 7.4. So when we do this in a preparative way, and when we have a look at this peak that comes out up front and look at its CD spectra, it is actually very different from the spectra we had at the beginning. Remember, this is kind of the shoulder where we have the structural content, but it's mostly kind of overtaken by a drop around 200, which is the lack of secondary structure content. But what we see here at this peak, which runs up front, it's a very, very pure beta sheet. It's actually a spectrum that you could find in textbooks, but you rarely see in your experiments. I have never seen one like that before. And when Misha, our student, sent this to me after the first experiment, I actually thought that it's a prank. It was not a prank. Repeated many times with different batches, we still keep seeing that with the 10 early alphabet or generally negatively charged alphabet based on early amino acids does is it forms these pure beta sheet species. This is just mass photometry showing the kind of size of this species, which is around 100 kilodaltons, but it's a whole broad distribution of molecular rates. So what we are observing here is not like a 5-mer, 6-mer, octamer. It's a bigger species. We now call it assembly, self-assembly of these peptides. When people see this beta sheet signature, the kind of usual association is amyloids. And amyloids have been vastly discussed in relationship to origins of life. This is one of the scenarios of how early proteins or protein-like structures could function using simple peptides. But the problem is that amyloids are always, not usually, always gained by sequences which are kind of identical. So it's one sequence sort of working as a template for another sequence in terms of structure and building up on it. I will just jump ahead. These are not amyloids. We did have a look at them under the microscopes. They do not form fibrils, and also their size is smaller. So what we are observing here it's not amyloids, it's actually soluble structures. Unlike amyloids, amyloids are very insoluble, yet they form this assembly, which even from very combinatorial sequences make up this very clean structure. We yet don't know much more about this, but what I can tell you from this part of my talk is that all the prebiotically tested non-canonical amino acids decrease secondary structure propensity, and that the 10 early amino acids somehow stand out, not just in their structure propensity, but also because of forming these higher assemblies. We think that these might actually kind of present stepping stones toward protein structure, because by forming an assembly, we are kind of bypassing the need of creating hydrophobic, or bypassing the mechanism of creating hydrophobic core if we just have short structures, if we just have short sequences. So the assumption is that you cannot actually have very easily hydrophobic cores and hence catalytic efficiency until you have long sequences of proteins which can fold into these structures. But we think that this is a sort of bypass by having peptides that do assemble that actually create these potential functional hubs. So this is something we are looking into, and could we actually associate some functions with these assemblies. So this is the end of the first part of the talk, and I'm gonna jump right away to the second part, which will be actually looking at protein-sized molecules, meaning longer sequences, which in terms of the time axis, I don't know if I had it here, no, I don't, which in terms of the time axis is already assuming we do have some form of translation or ribosomal synthesis happening. So we were again interested in, could these early amino acids actually give rise to some folding or compaction, or did we have to wait for the expansion of our protein alphabet to get those protein structures, those nice folds that we see? So first of all, we again compared the structure formation between our 10E amino acid alphabet and compared it to the full amino acid alphabet, which we here call 20F. Here we look at sequences which are 100 amino acids long. And first of all, we just look at secondary structure prediction. This is work done by Slava, who was the first PhD student here. And the first hint we got was that both 10E and 20F alphabets should give rise to similar structure content, similar alpha helical and beta sheet content. This was surprising. And at the same time, this was actually not true for any other set of 10 amino acids that we analyzed. This was something that, again, really kind of stood out among alternatives. So next, we decided to look at this experimentally, and Slava constructed a combinatorial library made of these compositions, made of early amino acids. This time we didn't do it by chemical synthesis, that would be too long. So this was done using cell-free expression, starting from a DNA template. And using cell-free expression, we could have about 10 to the power of 12 different sequences in one tube. What he did next was he looked at the resistance to proteolysis of these bulk libraries as a sort of approximation to protein folding. Remember, we have a huge variety of sequences. We couldn't do things like NMR or crystallization. So we're kind of left with very old school biochemistry. So we're exposing these sequences to proteases such as sorry, I forgot the name of the protease that we use, long protease. Again, we're looking at the 10E library, comparing it with the 20F library. And what's happening here, I will not go into detail, is that using this assay, we can actually differentiate both the degradable and undegradable species of the soluble and insoluble parts of the library. So these are the different shades of blue are the soluble undegradable in the darker and degradable in the lighter shade of blue, and comparing it to the 20F library. What is interesting, the first message is the 10E library is a lot more soluble. Compared to the 20F library, we see a lot of aggregation of these sequences. And what I would like to draw your attention to is the darker blue here, because this mostly resembles the proteins that you would like to see in your experiments, you would like to see in biology, proteins that are compact and undegradable, probably, and soluble, right? This is done in the absence of chaperones. Here, when we add chaperones, which today's biology uses to help solubilize proteins, we see that the soluble part of the 20F library expands greatly, while the soluble part of the 10 early remains the same, basically telling us that chaperones make a huge difference for the amino acids, for the alphabet that our life uses, but this was not needed earlier on when we just were having a restricted alphabet. So here we learned that the 10E alphabet kind of stands out among alternatives, and yet it doesn't require chaperones. But the next main question was, okay, could it actually support folding and function, which is something we could not really see from these combinatorial experiments. We actually had to go into specific sequences, into specific proteins to address that. The way we did that was using top-down approach. So we took some of the current proteins that we see in biology, and the palette of the colors here shows you the composition, the 20 different colors, meaning the 20 different amino acids. And what we were trying to do was simplify this palette, simplify the size of the alphabet into the 10 early amino acids. And we were interested in both enzyme catalysis and functions like RNA binding, which is very important in today's biology. And the task we are having here is we are actually trying to remove some of the most functional amino acids from these proteins, because if you look at enzyme catalysis, those amino acids which are most important here in the biggest font for active site activity is things like histidine or lysine, tyrosine, cysteine. Those are the major catalytic residues. And similar for RNA binding, we see arginine, lysine, aromatic amino acids. Those are all believed to be the late amino acids. So we wanted to see if we can still sustain these functions, even if we get rid of the amino acids, which today are most important for these functions. The first protein that we looked at was an RNA binding protein. It was a protein or a domain of a protein we borrowed from the ribosome, protein, the C-terminal domain of L11, shown here. It's a very small domain. And in green is the part of the ribosomal RNA that it binds to. And what Valerio, our postdoc here did was he looked at the sequence of this RNA binding protein, and every position which included the late amino acids shown here in blue, he basically offered a subset of amino acids from the early set. So he basically created again a combinatorial library which was based on the template, but each new position had a subset of options from the early set. And he did something called mRNA display, basically selecting from this pool of sequences for the one sequence which would still be able to bind RNA. And this was the starting sequence logos at the beginning. And this is what we end up with after about 15 rounds of the selection. What I would like you to observe mainly is that at the beginning, we had really a lot of positively charged residues, which makes sense. That happens a lot in nature when you bind to RNA. What we ended up at the end was that many of these sites were actually substituted by negatively charged amino acids, such as aspartic and glutamic acid. And this is what happened structurally speaking. So the original wild type had a lot of these positive negative interactions at the interface. What happened with our mutant, we have negative interface, but this was actually bridged by magnesium or potassium metal ions. So what we saw was that, yes, this interaction could help early on, even if positively charged residues were not around, but the positive charge was actually substituted by metal ions, creating a minus plus minus interaction instead of a simple minus plus. So we saw that, yeah, we can have protein functions using the early set, but we still didn't know whether we can have stable structures in the absence of negative or positive charge or aromatic amino acids. So this is the last piece of data that I'm going to show you. A very recent collaboration we had with Ingemar Andre from Lund University, and we decided we're going to look at some of the most ancient folds and try to redesign them to the 10 early amino acids. We did that using the very kind of modern protein design tools, using RF diffusion to basically just fix our backbone template, and then using protein MPNN to assign the sequence based on the 10 early alphabet. And I can just jump very quickly. We look at several folds. I'm going to show you one of the examples. Here, this is a ferredoxin-like fold, the wild type fold. It's actually functions as a metallochaperone, so it binds a metal ion, which is not shown here. And what you see in red is our mutant. Our mutant composed only of early amino acids, showing you nicely that the fold is almost copying the original wild type structure. So in conclusion, what I wanted to mostly stress is that the 10 early alphabet doesn't seem to stand out only at the sort of peptide level of things, but it can also perform some limited functions. We, of course, didn't look across the whole EC classes yet, and we are sure that it will have a lot of disadvantages compared to the full scope of the alphabet. But we see that it can bind to RNA. Our previous colleague Karo, who is here, also looked at the PDB analysis and saw that it can also bind organic and inorganic cofactors. And most importantly, and most recently, we saw that these early alphabets can support folding. Last slide, so I would like to thank the many people from our group involved in these projects, and also the rest of the group, and many of our co-workers at different places across the globe. And I would like to thank HFSP for funding most of this work. And thank you for showing up. I hope I didn't exhaust you too much. Feel free to ask anything, please.